My name is Sai Satish Mahadik. I am from SYBSc and my PPT is about exploring biomolecule structure, function and analytical technique. Biomolecule are organic compounds that are synthesized by living organisms that are very fundamental to our life process. Biomolecule plays an important role in maintaining the cell structure and integrity. It also helps in transmitting genes from one generation to another, also plays vital role in carry out many biochemical reactions. Biomolecules are majorly classified into four forms, namely carbohydrates, lipids, nucleic acids, and proteins. Nucleic acids are linear unbranched polymer composed of monomeric unit called as nucleotides. Nucleic acid was first discovered by Friedrich Mischer. He also isolated a substance called as nuclein, isolated from WBCs. Later, that substance was recognized as nucleic acid. Nucleic acids are divided into two parts, DNA and RNA. DNA stands for deoxyribonucleic acid. It is a double-stranded structure which is present in the nucleus. It contains deoxyribose sugar and it helps in process of transcription. Second type is RNA, which is ribonucleic acid. It is a single stranded structure present in nucleus and cytoplasm. It contains ribose sugar and helps in process of translation. This is the structure of DNA and RNA. Now, the importance of nucleic acid in various cellular and metabolic activities. Nucleic acid plays a very important role in genomic stability. It stores and transforms the genetic information from one generation to another. Along with that, it also helps in protein synthesis, in which RNA translates the genetic code from DNA to make specific proteins through transcription and translation. It also supports in cell division, that is, DNA replication ensures that each new cell has an exact copy of genetic material during meiosis and mitosis. Now, this is the composition of nucleic acid. Nucleic acid is composed of nitrogenous base, pentose sugar and phosphate group. Now, the nitrogenous bases are majorly classified into pyrimidines and purines. Purines are large two-ringed molecule containing adenine and guanine. Pyrimidine is a small and single ring structure, contains cytosine, uracil and thymine. Uracil is present only in RNA and thymine is found only in DNA. Now, the functional groups that are present in nucleic acids. Nucleic acid contains amino group which is present in the nitrogenous base, hydroxyl group which is present on the pentose sugar, and phosphorus atom which is bonded to oxygen. The forms of nucleic acid. Structurally, nucleic acids are classified as primary, secondary and tertiary and quaternary structure. Primary structure contains a linear sequence of nucleotide monomers bonded by phosphodiester bond. The secondary structure involves local folding patterns like double helix in DNA and with complementary bases. RNA includes a stem loops and folds back to itself. Tertiary structure refers to 3D folding of nucleic acid molecule. It is more complex than the other two. Now, the role of nucleic acid in energy carrier. Nucleotides act as primary source for between the phosphates. The second is chemical messenger. Apart from their role in energy transfer, they also act as signaling molecule. They also transmits hormonal and extracellular signals. Also components of enzyme, cofactor, etc. Presence of adenosine gives a molecular handle that enhances the binding affinity between the enzyme and cofactor. Now, the methods that are used to study nucleic acid. First is PCR, that is polymerase chain reaction. It is the method relies on repeated cycles of heating and cooling to amplify the DNA fragments. This method creates millions of copies from a small sample. This method is used in disease diagnosis and forensic analysis. This method includes denaturation, that is, the double-stranded DNA is heated to separate the strands. Second is annealing, primer binds to the complementary bases and on the single-stranded DNA. Third is extinction, Taq polymerase extend the primer synthesizing the new strands of DNA. Second method that is used to study DNA or the nucleic acid is the DNA sequencing method. This method is also called as NGS, that is next generation sequencing. This method is used to determine the order of nucleotide in DNA molecule. This method includes steps such as sample preparation. The desired biological sample is prepared. Second is fragmentation. The large chains of DNA are broken down into fragments. Then the primer annealing, in which the next step is primer annealing, in which the short single strands binds to the complementary DNA template. And the last process is DNA synthesis, in which DNA is synthesized and later analyzed. The last method to study nucleic acid is gel electrophoresis. This method is used to separate and analyze the molecule like DNA, RNA or proteins on basis of their size and charge. This method includes gel preparation, that is, a gel is prepared with specific concentration of agarose and a comb is used to create wall where the sample is loaded. Then second step is sample preparation. Molecules to be separated are mixed with loading dye. Then electrophoretic run. The gel is placed in the gel box filled with buffer and electric field is applied. Visualization after the gel is stained. After this, the gel is stained to make the separated molecules visible. DNA is stained by is visualization, in which the gel is typically stained to make separate molecule visible. DNA molecule can be visualized using dye like ethidium bromide under UV conditions, while proteins can be visualized by using various protein stains. Then the next step will be analysis. The separated molecules appear as band in the gel, and their positions can be separated separated and analyzed to determine their size and relative abundance. Now these are the references which I used to make this PPT. Thank you.